presentation is about regenerative medicines. This was made by Quinn, Nour, Yasmine, and Kenzy. Here we have a vocab sheet with some definitions you need to familiarize yourself with in order to understand the struggles these scientists went through when studying regenerative tissue problems.
Autologous means to be taken from an individual's own tissue, cell, or DNA. Fibrosis is the thickening or scarring of the tissue. Cell differentiation is the process during which young, immature cells take on individual characteristics and reach their mature form and function. Retrovirus is a virus that uses RNA as its genomic material. Upon infection with the retrovirus, a cell converts the retroviral RNA into DNA, which in turn is inserted into the DNA of the host cell. Pluripotent stem cells is a cell that is able to develop into many different types of cells or tissues in the body. Grafts, healthy skin, bone, or other tissue taken from one part of the body and used to replace diseased or injured tissue removed from another part of the body. Systemic immunosuppression is the state in which your immune system is not functioning as well as it should. Induced proliferation is multiplying or increasing it unnumbered.
In this scientific journal, scientists want to create medicines and therapies that can resolve regenerative issues in a person's body. More than 45% of deaths are caused by inflammation and fibrosis, which is why scientists are currently exploring the challenges regenerative medicines bring to the lab. A key question that is yet to be answered is how can human tissues be persuaded to regenerate? The article focuses on the prevention of poor cell differentiation, fibrosis, and chronic inflammation because these diseases have solutions to them that also bring along unresolved hurdles. Facing these challenges and finding ways to unravel the truth behind tissue regeneration can make its way to modern-day medicine. A requirement that must be met in order to find a solution to their question is to replace the damaged tissues with those of the wild-type trait. For example, a child with a lethal skin disease caused by a mutation of his gene was treated with the use of retroviruses. It will be replaced with the wild type that the retrovirus carries. When the cells undergo mitosis, it will create more functional cells that will be used in a graft. Another therapy involves using stem cells to act as needed molecules the person may be lacking. This is used to reduce the symptoms of diseases such as type 1 diabetes and Parkinson's disease. Using grafts is a perfect way to enact this type of therapy. However, it comes with its downsides.
Issue 1. Immune system rejecting grafts. The goal of regenerative medicine. In order to treat the damage or the loss of stem cells in the body, regenerative therapies must be able to replace what was lost. One method used to achieve this goal is the use of invigorating endogenous stem cells, using stem cell based therapies including grafts. Case study. A young boy suffering from epidermolysis bullosa was treated with a retrovirus that transduced the wild-type laminin gene responsible for its condition. Healthy tissue was grown from an autologous tissue, expanded, and then attached to the damaged parts of his skin as a graft, which restored almost 80% of it. Early benefits. Out of the treatments listed, stem cell based therapies have shown the most promise within clinical trials. Human pluripotent stem cell therapies have entered clinical trials for various diseases in which one type of the cell is needed to be repaired for treatment. Here are some examples. In type 1 diabetes, insulin is not produced enough. In Parkinson's disease, it's MDA neurons. In age related macular degeneration, for retinal epithelial cells. For example, a Parkinson's disease patient who received treatment within his midbrain showed reduced symptoms and improved motor functions over a 2-year period. Hurdles in treatment. Efficacy is best achieved through the use of autologous stem cells for grafts as they avoid immune system response through recognition as native cells in the body. The drawback is the high cost labor intensity, as well as extensive safety testing of this type of treatment. It's not as accessible as other forms of treatment. Use of non-autologous stem cells is currently being researched, but it's shown drawbacks as well, with the most major being immune rejection of non-native cells, which lead to damaged grafts. Alternatives. Long-term use of systematic immunosuppressants, encapsulating the graft with a selectively permeable membrane, rejects immune components, accepts nutrients, using alginate encapsulation to deliver cells. A clinical trial for delivering factor VI5 cells for hemophilia treatments led to adverse immune responses. The capsule was found to be fibrosed. Non-autologous treatments still need to be developed for accessibility and functional purposes. Issue 2. The biggest issue posed with cell therapies is the growth of cells either too rapidly or too slow. When it grows too rapidly, this often leads to tumors. Stop growth signals are not well understood enough to help stop abnormal growth. Lastly, it was believed that the hypnosignaling pathway was the pathway that controlled this over or undergrowth, but recent studies found it is unclear to determine if separate pathways control regeneration in adult tissue. Issue 3. Another issue faced is the integration of grafts into the corresponding parts of the body. The body struggles receiving signals to communicate with the nervous system and blood cells. Scientists' solution to this is to reprogram the cells at the damaged site to become the needed type of cell. This issue with this method is with its long exposure, this usually led to development of tumors. Lastly, it was noted that in zebrafish, their bodies only activated reprogramming in response to the stimulus of an injury. Scientists aim to find a way to use the same method in humans.
Issue 4. Inflammation is the body's disease, injury, illness, and bacteria and plays a crucial role in the body's overall health. When inflammation happens, when there are no harmful invaders, it can damage the body's health, and this is called chronic inflammation. Regenerative medicine is looking to solve chronic inflammation using stem cells to regenerate body tissues. Chronic inflammation causes fibrosis, which is a thinning or scarring of the tissue caused by chronic conditions like chronic inflammation. The ongoing irritation: when the body faces constant sickness, it will constantly be facing inflammation, which causes too much fibroblasts. Fibroblasts are a type of cell that makes new connective tissues, and much of these cells being produced causes and destroys organ functions, leading to scarring of tissues. This will cause too much ECM, which is dermal extracellular matrix. Too much ECM leads to the body tissue to becoming thicker, leading to scar tissue, which affects the function of the organ tissue and joints being affected. It also can cause inflammatory bowel disease, which is a result of a weak immune system. The solutions are stem cell therapy, which are stem cells are specialized cells that have two properties. They are able to make cells just like them, and through differentiation, they can become other cells. Stem cells are taken from a donor through a process, and then the stem cells are taken and processed in a laboratory to isolate them. The stem cells can be given to a patient through an injection. We also have synthetic psychokinesis, can induce signaling patterns. These cells are small proteins that control the growth of immune cells and release their signals for the immune system to do its job. This mimics the process of cell division, but in a controlled manner also. Genetic engineering is used so the cell division can be controlled. This is done by making synthetic genes into cells and that can respond to specific signals to control the amount they divide. We also have antifibrotic therapies. Antifibrotics are drugs that inhibit fibroblast activation, which causes inflammation. Antifibrotic therapies can also lessen EMC, which causes fibroblasts, and the stiffening of the joints of tissues by increasing matrix multiplicasis, which decreases EMC.
Now that you've gotten a better understanding of how scientists are trying to find ways to combat regenerative tissue failure, let's look at the pros and the cons. The grafts help repair or replace lost tissue, so when the cells on the graft undergo replication, it can replace most of the missing tissue. However, some cons of using a graft is the immune system might reject the graft, which can either lead to the cells being destroyed or causing inflammation. However, scientists did find a way to prevent this from happening, which was to add a selectively permeable wall, which doesn't allow immune cells to come through. However, grafts are not able to go in certain places because it's difficult to apply a graft there, so scientists decided to change the transcription factors in order to solve the issue of regenerative tissue failure, but that only causes the production of tumors if not monitored. While there are some therapies and medicines, including inhibitors, that can help with symptoms, it's not a permanent solution. There are still so many unknowns when it comes to regenerative tissue failure that is yet to be discovered in order to fix this problem.
Bioconnections. This topic of cell regeneration largely relates to Units 4 and 6. Unit 4 is the study of cellular communication. In the issue we looked over, we saw if the body rejected the graft, this would cause inflammation due to the body being unable to communicate with this new tissue and initially rejecting it through the use of NK cells. Unit 6 was the study of gene expression and regulation. One of the issues faced was integration of a graft. The solution was to reprogram the cell or tissue by changing the types of transcription factors, allowing a new type of cell to be encoded for.
Thank you for watching our video. We hope you enjoyed, and we hope you learned a lot about regenerative medicine.